Hi everyone, I'm Nicolas Benoit, co-leader of this collaborative project called Ring of Fire, Reconstructing long-term environmental records to support regional assessment. This is the third annual presentation for this project. To avoid repeating myself, I leave you here as a reference the links of the past presentation project in case you are interested in more background. In this project, the main objective is to improve the knowledge on the evolutions of the wetlands baseline conditions in response to climate and anthropogenic stresses and their effects on metallurgists' mobility. Chromite mining activities in Ring of Fire could be a source of chromium mobilization in the environment. Why are we interested in chromium? Because there's a gap knowledge about the fate of chromium species in this context. Total chromium in the water is a mix of different chromium species concentration, where chromium-3 is essential for health and involved in various body processes, rather than chromium-6, which is highly toxic. Chromium species are difficult to measure in water because they are affected by field, sampling, and analytical conditions, making it difficult to obtain representative results. We have two general activities objectives. The first one is to propose efficient chromium speciation methodology for sampling and analysis. And the second is to define the chromium-3 and chromium-6 species occurrence in post-mining context of chromite deposit at Ried-Bélanger and Montreal mines. Today, I present only the water geochemistry aspect, but we have a multidisciplinary teams working on other aspects of the project, such as the mineralogy, organic soil, wetlands, and lake sediment. Okay, this is the study area. On the map on the left, you can see the location of the Ried-Bélanger mine here, the red rectangle with an A, and westward, Montreal mine in the B rectangle. On the right, is a knowledge view of each mine. These mines are located in the aphylitic complex of Thetford, mainly composed of ultramafic and mafic rocks. Ried-Bélanger, which was the most important chromium mine in the region, have a vast staining area, as you can see here on the map and on the picture. On this side, there's also several small pits here and underground galleries. The Montreal mine has the largest open pit with extensive waste rock area to the southeast that divide nearby wetlands. The pink and the green points, the map indicates the location of surface and groundwater sampling sites since the beginning of the project. Those indicated by the arrows were revisited last September. Okay, here we have some geochemical background. For our site, the pH values suggest a neutral to alkaline water. pH varies significantly for Montreal mine between 7.4 to 9.8. Ried-Bélanger is particular with a very high pH around 10 for our sites. And surface water outside of the mine is around 8. On the right, the TDS measure are representative of hard to very hard water for mines, and it's a bit less hard for streams. The Piper diagram shows an absolute dominance of magnesium bicarbonate. All the points are clustered here. This water type is representative of recent research and also ultramafic bedrock interaction. Here, it's difficult to read, but the diagram supports this water-rock interaction for mines samples. This diagram also shows that streams located outside of the mine are somewhat more influenced by precipitation. For chromium analysis, we focus on chromium-3 and chromium-6 separation using frontal chromatography. In theory, the role of the frontal chromatography is to retain chrom-3 and let chrom-6 pass through a cartridge. This cartridge is filled with a strong cation exchange resin, blocking cationic and natural chrom-3 and leaving anionic chrom-6 as the only unretained species. Chrom-3 is calculated as the residual of total chromium minus chrom-6. Chromium species concentration was measured by ICP-MS. As you can see on this graph, the standard calibration shows good separation between the two chromium species. For development and validation, all samples were collected twice and analyzed in the GSC Ottawa laboratory and Brooks Applied Labs. We first look at total chromium. For this analysis, some samples were spiked by adding 10 ppb of chromium-3 and chromium-6. The spikes help to measure the evolution of the species between the sampling and the analysis. For all samples, the detection limit of the analysis is 0.02 ppb. These graphics here compare non-spiked top and spiked sample concentrations, just below. For unspiked sample, the maximum concentration is 4.18 ppb. Spiked samples show low concentration. Indeed, expected value for spiked samples should be greater than 10 ppb. This result suggests that there is a potential recovery issue. Okay, now we look at the recovery result for sample spiked with chrom-3 and chrom-6. What is important in this table are this column here and this column here, which correspond to the spike recovery for chrom-3 and chrom-6. In this column, values in red correspond to problematic sample, where 3 were over-spiked and 2 were not spiked. The result here for the chrom-6 show good recovery at over 90% for all samples except 1. Similar results from Brooks Lab are a positive indicator that we can confidently discriminate chrom-6. It is a little less good for chrom-3. There's probably a pH effect here, where chrom-6 tend to stay more stable at high pH. There is also a possibility for co-precipitation of chrom-3 and accumulation on bottled in a wall, causing low recoveries, but further tests are coming for this hypothesis. But organic matter is probably the most important factor here affecting the results. This year, we spiked before doing the frontal chromatography. We hope this will improve the results and provide additional information regarding frontal chromatography efficiency, in particular regarding the impact of architectic power. There is a strong linear correlation with total chromium and use of organic carbon, where we expect possible chromium-3 organic enrichment. For high DOC value, results show a frontal chromatography pulse post some form of chromatography can combine with organic matter and give as an end, and therefore can pass through the cultural action to give a pulse positive form of 6. Organic matter is probably playing a major role in this system by mobilizing chromium, and if it's the case, this is an important finding of the study. For low DOC values lower than 2 ppm, frontal chromatography spikes or not, Chrome-6 show similar results. Organic matter could have unpredictable effects on both speciation and chromatographic separation. As mentioned before, with the recent sampling, we will make new experiments to better understand the organic matter effect. So we conducted a new field campaign last September for which we adapt field methodology to resolve some 2021 issues. Spice concentration were reduced from 10 to 2 or 5 ppb to be more representative of material concentration found at the site. Faster shipping analysis was done to reduce interspecies conversion impact. New in-house experiments and internal laboratory comparison continue to validate the development methodology and better understand the organic matter effect. And this year, we also sampled some isotopes to better understand the water regime and ultimately the mobility of the chromium. And that's it for me. Thank you all.